Imagine waking up with a headache, so intense that even the slightest movement feels like a hammer pounding in your skull. You can't see it, but it's all consuming. This is what millions of people experience every day, yet it's a condition that often goes unnoticed by others. I'm talking about migraines. For those who don't suffer from them, it's easy to dismiss a headache as just a minor inconvenience. But for those who experience migraines, it's a condition that affects every aspect of their life. Hi, my name is Keerthana Sridhar, and to share a little bit about myself, I'm presenting from Ontario, Canada, and I'm a junior at Bill Hogarth Secondary School. My research has been guided by my mentor, Miss Natalie D, and additionally, I have a passion for biomedical engineering and wish to pursue it in the future. For me, this research is more than just a project. My grandmother, my mother, and I have all been affected by migraines. So I've seen firsthand how the impact it can have on one's life. This personal connection is what pushes me to dig deeper into the genetic causes of migraines. By understanding the genetic factors behind migraines, we could make steps towards developing better treatments, not just for my family, but for the millions of others who suffer in silence. So, what exactly is a migraine? Migraines are far more than just a headache. They're a neurological condition that affects over 1 billion people worldwide. For some people, it includes symptoms like dizziness, vertigo, and nausea. This type of migraine is called vestibular migraine, and it impacts the brain and inner ear, making it difficult to maintain balance. They can last for hours or even days, significantly disrupting daily life. Migraines like this are not well understood, which is why studying the genetic causes is very important. Now, into the genetics component of migraines. Did you know that up to 60% of migraine cases are believed to have a genetic component? But here's the problem. While we know that genetic plays a role, finding the exact genes responsible for migraines, especially vestibular migraines, is still a mystery. In fact, look at this autosomal dominant inheritance pattern. If one parent has migraines, there's a 50% chance the child would inherit them. But this question still stands. What are these genes and how do they work? That's what my research aims to uncover. To investigate these genes, I used a genomic data set that included 6 vestibular migraine cases and 6 healthy controls. I applied RNA sequencing and bioinformatics tools to analyze the gene expression. This allowed me to find genes that were different between migraine and control groups. The key steps in this process included alignment of raw RNA data using Callisto, differential expression analysis with DESeq2, gene pathway analysis using GSEA, and these last 2 steps were done with the coding language R. While I have large tables and figures with gene pathways and genes, I'll be talking about the most significant of my findings. One of the most significant findings was the upregulation of a gene known as CARS-S1, which means increased upregulation means increased activity in a person. It is important in protein translation and mitochondrial function. Mitochondrial dysfunction is a known cause of migraines because it affects how brain cells utilize energy. The CARS1 is hyperactive in migraine patients, indicates that brain cells of migraine patients might be having problems in energy balance, and this could be part of the migraine mechanism. Further investigation of CARS1 might inform us more on its role in migraines. Conversely, I found that the most downregulated gene was RSAD2, which means its activity is inhibited. This gene works to inhibit viral infections and regulate inflammation in the brain. If this gene is less active in individuals who experience migraines, it could indicate that their immune system is not keeping inflammation under good control. Recognizing that neural inflammation is a key migraine trigger and determining how this immune dysregulation plays a role may uncover new therapies. Now let's look at the gene pathways. One of the most upregulated pathways was found to be WP phosphodiesterase in neuronal function pathway. This pathway controls the enzyme phosphodiesterase 3, PDE3, which breaks down molecules such as cyclic AMP and GMP. AMP and GMP are molecular messengers that are vital to controlling neurotransmitter release and communication between brain cells. If this pathway is disrupted, it could interfere with the proper communication of brain cells. If this pathway is also disrupted, it can potentially lead to migraines. Interestingly, in other studies, PD3 inhibitors have been used to reduce the severity of headaches in some conditions, suggesting that this pathway is already a target for future treatments. Next, I found that the KEG riboflavin metabolism pathway was the most downregulated in migraine patients. Riboflavin, or vitamin B2, plays a crucial role in mitochondrial function and energy production. Since migraines are often linked to mitochondrial dysfunction, the underactivity of this pathway can contribute to the energy deficits that migraine patients experience. Additionally, riboflavin supplementation in research papers has been shown to reduce the frequency of migraines, which presents the importance of this pathway in migraine development. So, where do we go from here? To build on this research, we need to expand this study, increasing the sample size and including more diverse populations would help validate these findings to make them more generalizable. Future studies should also explore immune pathways in migraine progression and focus on understanding the genetic mechanisms behind vestibular migraines. By identifying more genetic markers, we can develop more targeted therapies tailored to specific genetic profiles. In conclusion, migraines are a complex condition influenced both by genetic and environmental factors. My research has highlighted key genes like CARS-S1 and RSAT2, as well as pathways that like PDE3 and riboflavin metabolism, which could help lead to new treatments. The next step is to explore how these findings can lead to better therapies, especially through immunomodulatory approaches. Thank you for your time, and I'd be happy to answer any questions.
Fantastic. Thank you so much. One question I have for you is, how has doing this project kind of changed your relationship to your own migraines or kind of changed the experience for you when you get a migraine?
Well, when I get migraines, I feel like I have always seen them as something that hurts, but I've never wondered why it hurts, and that definitely drove me to do this project. And especially that this project tells me more about the genes that might work
Mhm.
behind these migraines, and especially the pinpointing exactly why, with the riboflavin metabolism, like the vitamin B2, like how that might be deficient in people, or the other things like the PD3 inhibitors. So that just gives me more information how there's so many small proteins and genes that work for me to have to experience this condition. And yeah.
Awesome. I love it. Um, there's someone in the Q&A who says, "Great presentation. How did you find the data set you used?"
So I found this data set on the NCBI database, and it was actually the one of the only, I think it was the only migraine data set I could find actually. So it shows that how less these gene sequences for migraine patients are there. Yeah.
Yeah, gosh, I would expect a lot more. That's really
Yeah.
interesting. And that feels like, something other folks have brought up in this panel too, of just like needing more attention on a lot of these different issues too. Amazing. Well, thank you so much, Keerthana.
Yeah, thank you.